Okay, today we're going to start talking about chromatin and transcription, starting with stuff you kind of remember from Cell and Molecular and moving off into slightly more uncharted territory. Okay, first I want to go over a little bit about the worksheet that we assigned. Here's basically the answers. There's no real trick to this. Just follow the directions carefully. The only hard part is sometimes you have to write sequences backwards in order to be able to locate their complements. The little blue thing on the picture represents the scaffold part of the guide RNA, and the part that I've typed is the part that's the actual guide sequence. Sorry, I've got T's instead of U's, but it is actually an RNA. Okay, same with this one. The second one's a little bit harder because the sequence you're starting with does not include the PAM. So you're going to write the sequences of the guide and its complement, and then you need to align the guide to the complement in the usual way. And the problem with this is that the guide does not include the PAM, so you need to go back to the genome sequence to find that and locate it on this. But just struggle with it a little bit. It should not be difficult once you kind of figure out where to align things. Okay, meanwhile, we're going to talk about eukaryotic gene structure. Here's kind of a stripped down version of a eukaryotic promoter. Notice that it's not to scale. It's got a humongous promoter proximal region, which is really only a few hundred base pairs. And the gene itself, which is in red, is very small compared to the real gene. Notice that the red arrow moving to the right, that represents the transcript start site, where polymerase will actually start copying the template. And in blue, we have the Tata box, which is within, in this case, 30 base pairs or so upstream or before or 5' to the transcript start site. So, of course, the Tata box, if it's there, is where polymerase will eventually assemble with the help of a host of transcription factors, etc. The promoter proximal region will bind proteins that can be either activators or repressors, which either facilitate or prevent from binding RNA polymerase. The enhancers, of course, can be much further away from the actual protein coding or transcribe region of the gene, as we'll see in a little while. So here's one representation of RNA polymerase trying to get itself to the Tata box. You can see that it itself does not bind directly, but actually occurs with a whole host of transcription factors. So the TF2s refers to transcription factor for polymerase 2, and they're given letters. And each one of those is actually a protein complex in and of itself. We also have the mediator, and we're also representing activator proteins that are bound to regions within the proximal promoter region that are going to facilitate assembly of the pre-initiation complex at the Tata box. Taking into account our enhancer, this is from the Cell and Molecular book, we are showing RNA polymerase, which is bound to the promoter, is about ready to get started, along with mediator, other transcription factors, but importantly, activator proteins that are bound to the enhancer, which is quite far away from the transcribe region of the gene. So we're representing this as a DNA loop. In fact, it is true that enhancers can form loops, the DNA forms loops to bring what would be really quite far away in the DNA to the vicinity of the promoter and bringing proteins and sometimes RNA polymerase along with it. We'll have more to say about these at some later date. So general terminology here. Cis-acting sequences refers to the sequences that are on the same molecule. For example, the Tata box is a cis-acting sequence because it controls transcription that will occur on the same DNA molecule as itself, but not somewhere else in the genome. Proximal promoter elements are also cis-acting, enhancers, etc. The proteins that bind there are considered to be trans factors because they come from somewhere else and can act on different molecules from themselves. So that's just one way of just thinking about DNA sequences and the molecules to which they bind. Okay, importantly, we realize we've got to get a lot of proteins there, but we've got to deal with the nucleosomes in the process. So the accessibility of these sequences depends greatly on the configuration of the actual chromatin and the organization of nearby nucleosomes. So we're going to need to be able to get all these proteins there, but we sometimes have the problem that the nucleosome is in the way. So it's very important to get the nucleosome out of the way to get transcription to occur. So we can't bind if there's proteins in the way. Most of the time, promoters are hidden from RNA polyp II by nucleosomes. Here's a picture of chromatin with the nucleosome core particle shown. The nucleosome core particle consists of an octamer, or 8 proteins, 2 molecules each of histones H2A, H2B, H3, and H4. And the DNA is wrapped around the nucleosome core octamer, approximately taking up about 146 to 147 nucleotides of double-stranded DNA. But notice that there's part of the DNA which is not actually covered by the nucleosomes. Those are the linker regions of the DNA, and those are going to be important in terms of getting access to the DNA sequences that the proteins are going to bind. So I think I said this, H2A, H2B, H3, and H4. On average, it's about 200 base pair per nucleosome, but this is not always constant. We've got the DNA that's wrapped around the core, and the part that's not wrapped around the core is the linker. There's also histone H1, which may also be bound to the outside of the nucleosome. So the term chromatin refers to these complexes of proteins along with the DNA. Okay, shown here is a detailed view of a nucleosome viewed from the top. You can see that the DNA is wrapped around the protein core. But importantly, the proteins have extensions, the N-terminal tails, which protrude out from the nucleosome and are available for action by various modifying enzymes such as acetyltransferases and methylases, etc., as we will see. So they are exposed to the environment. Okay, so in terms of allowing access to the chromatin, there's differing states of chromatin which will depend on the types of modifications that they've undergone. For example, we'll have modifications of those histone tails that are sticking out, acetylation and methylation mainly but not exclusively. There's also DNA methylation. We also have to consider actions of chromatin remodeling complexes. And access, of course, is also dictated by the degree of compaction of the nucleosomes into higher order chromatin structures. We'll do the tail modifications first. So here's a picture that kind of represents that, where you have part of the chromatin which is nicely strung out, linkers available, nucleosomes available, a nucleosome-free region, but then a region where the nucleosomes have formed some higher order, more complicated structure in which the actual DNA is much less available than is the open structure. Yet another representation of the same idea, that genes that are actively being transcribed, the DNA is much more available than it would be in a compacted state. And there are proteins and complexes which dictate which form the chromatin takes. We're going to talk about acetylation and deacetylation in a second, but the acetylated forms of the histones are associated strongly with activated transcription, but that is a reversible process. There's also forms of chromatin where the acetylation hasn't really formed yet, but it's nonetheless accessible to get that process started. But there are also various forms of repressed chromatin which do assume higher order compaction, and that's associated with largely methylation, but also cytosine methylation of the DNA. Making a long story short, the transcription possible form has accessible DNA, relatively speaking, because it's got acetylated histones, its cytosine DNAs are not methylated, RNA polymerase can bind. The switched off version has condensed chromatin, deacetylated histones, and methylated cytosines, among other changes. So we're going to depend on various histone modifications and the proteins that create and read these. Okay, so we're going to be looking at the N-terminal histone tails, those that extend outward from the core. Okay, so we can modify these in ways that lead to opening of the chromatin or repression of it. Acetylation is associated with accessibility. So acetylation allows access of proteins to the DNA. HATs, or acetyltransferases, do this, histone acetyltransferases. Okay, this is a reversible reaction and a very dynamic one in cells, where the reverse process is catalyzed by H-stacks, or histone deacetylases. Okay. Methylation is not a plus or minus thing. Some methylations are involved in activation, others for inactivation. So whereas acetylation is almost universally associated with activation, methylation can go either way. There are other modifications that are also possible that we won't talk about specifically. One is phosphorylation, of course, and the other is ubiquitination. Okay, so H-stacks and HATs add or remove acetyl groups from the lysines and arginines. And I'm going to show you what that process looks like in terms of the actual amino acids. For example, here's histone, human histone H2B, just as an example, where I've colored in red all the lysines and all the arginines. These are the basic amino acids which are potentially subject to modification, either by methylation or by acetylation. Okay, so it's a very basic protein, as you can see, because it's got a lot of these lysines and arginines, mostly lysines. Here's lysine and arginine, and we're talking about the amino group that's part of the side chain. These both have positively charged amino groups as part of their side chains, and they are basic amino acids because they are positively charged at cellular pH. So the action of this, you have to turn your amino acid upside down now. The amino group is now located at the top. This is lysine. Acetylation by HATs, histone acetyltransferases, involves adding a 2-carbon acetyl group to that amino group that's part of the side chain, the epsilon amino group. So that removes essentially the positive charge from that side chain and allows for a looser organization, or a less stable, shall we say, nucleosome at that position. This is, of course, a reversible reaction, which is accomplished by the H-stacks. The other important modification is methylation, also of lysine, and there's 3 different types of methylation you can have, again at that amino group on the side chain. One is you can have monomethylysine, you can have dimethylysine, or trimethylysine, and these reactions are characterized, or catalyzed rather, by histone methyltransferases, abbreviated HMTs. Now, some of these have their own names. The HMT is just a generic term for the histone methylases. Okay, so it's very prevalent on basically all of them. The most important ones, most extensively modified ones rather, are the H3 and H4 tails. H2A and H2B can also be modified, but are also likely to have ubiquitin added to specific lysines. Okay, ubiquitin is a 76 amino acid protein, so this is a huge, enormous, actually, addition to histone protein if you consider the histone protein itself is less than 200 amino acids. Okay, so remember, some amino acids can be acetylated or methylated depending on the situation. We've already said this. Histone acetylation is associated with active genes, open chromatin, high levels of gene expression. Okay, some types of methylation are associated with repression and others with active transcription. For example, histone H3 lys 27 methylation and H3 lys 9 methylation are associated with repression. Okay, so here's a picture of H3 and H4 at the potential sites of methylation, acetylation, and also phosphorylation. This slide is not easy to look at because they've got red and pink, and it's hard to tell which one they're talking about. So I've got a better picture in a second. Okay, here's a histone H3 tail, and the colors are much easier to see. And you can see that some amino acids can be modified in different ways. For example, lys 9 can be either acetylated or methylated. Similarly, lys 27 can be acetylated or methylated. And these modifications, which do not occur simultaneously on all the amino acids, obviously, have different meanings. As we said a minute ago, the lys 9 methylation is associated with gene silencing, and the acetylations are associated with gene expression in general. So many different modifications and many different patterns are possible according to the gene, and the combination of what's modified changes from gene to gene and from activation state to other activation states or inactivation. They're very dynamic modifications usually. We've said this before. HATs, histone acetylase, histone deacetylase, histone methyltransferases. Okay, one thing that's important to recognize at this point is these enzymes aren't just floating around independently. They're generally part of large multi-protein complexes. And this goes for the acetylases as well as the methyltransferases. I don't think I mentioned this in class, but I will next time. There are repressor complexes, PRC1, PRC2, as an example of something that's part of a much larger protein complex developed, and that's associated with repression and silencing. So here's DNA methylation, that this is associated with repression, where the cytosine is modified to form 5-methylcytosine. And finally, we have chromatin remodeling, which goes hand in hand with our histone modifications. And there are different pathways by which a gene or genetic region can be activated. There's no simple linear process whereby this occurs. Okay, in one important way, there are chromatin remodeling complexes, ATP-dependent chromatin remodeling complexes, which actually act directly on the nucleosomes. They can slide them along the DNA, and they can evict them or disassemble them. The modifying enzymes can actually occur as a way of recruiting the chromatin remodeling complexes. Alternatively, you can have the action of the chromatin remodeling complex followed by recruitment of acetylases, for example. So there's no simple linear pathway that describes all this. So these are, in some sense, independent processes, and yet they are mutually dependent in many cases. But they are fundamentally different because the remodeling involves moving the nucleosomes around in some way, whereas the modification on the enzymatic modification by acetylation or whatever that's basically just a modification of that particular lysine on that particular nucleosome. So chromatin remodelers, there are bunches of these, and they work in different ways. The most famous one is one called a protein complex called SWI/SNF, and this one can eject or slide nucleosomes. And these generally are activators in transcription, usually acting in the promoter region. Okay, this has a human homolog, and it's called BAF in humans, or else BRG1. So it's actually identical, well, not identical, very similar complex in the yeast versus the human, but they were discovered independently, so they have different names. Okay, but there are remodeling complexes which work in reverse that are associated with repression, and these may contain HDAC or HMT activities as well. Okay, so now we need to consider promoter architecture and the formation of nucleosome-free or nucleosome-depleted regions, and the issue of nucleosome positioning. So this slide shows a picture of a yeast gene called FO5 in its repressed state. And the +1 nucleosome is shown at the beginning of the region that will be transcribed. The TATA box region is occluded by a nucleosome. The TATA is covered up by the fact that there's a nucleosome there. So in response to inducing conditions, a lot of stuff happens, which is very schematically shown here. But the bottom line is that remodeling complexes and histone acetylases will remove those nucleosomes that are adjacent to the +1 and allow the binding of the transcriptional activator, which is FO4. So this nucleosome depleted region is very characteristic of active genes. And we need protein complexes, etc., to get them that way. So here's a schematic diagram of sort of a canonical promoter, okay, where you've got the +1 nucleosome, and then there's a nucleosome-free region, which includes the TATA box. The little arrow on the +1 nucleosome indicates a position where transcription will start, and everything else there is proteins that are bound in the vicinity. We've got Pol2, mediator, and the little E and F things are the TF2s. The rest of the proteins are just shown sort of schematically. Those are the names of some of the protein some of the chromatin remodelers, which come into play when we're trying to get our gene activated. Some of the other proteins shown at the bottom are specific proteins, not protein complexes, which bind to DNA sequences in order to enhance mostly enhance transcription. So what this shows is a gene, a theoretical gene, with positioned nucleosomes. So what's being represented is each nucleosome, numbered 1, 2, 3, etc., and what the peaks are is the center of each of those nucleosomes. The idea is we will have a transcription start, and it's going to make progress through the gene, but the nucleosomes occur in this particular gene in a very ordered array and with equal spacing. Right, so you need to have a nucleosome-free region, or at least depleted region, to begin. And one way in which this can happen is that some DNA sequences do or do not favor the formation of nucleosomes. So there are sequences often within promoters that basically reject nucleosomes, where nucleosomes don't sit very comfortably. And so to some extent, nucleosome positioning is dependent on the underlying DNA sequence. Most promoters, however, need help to get the nucleosomes out of the way, even if they do have some sequences that discourage nucleosomes from forming. Okay, so we need to get the nucleosome in a position so that the +1 nucleosome is prepared for the usage of the correct transcription start site. Now, there's various proteins and protein complexes that are involved in this process. One type is general regulatory factors, and these are going to be mostly sequence-specific DNA-binding proteins that have their own specific recognition sequences. But there's also the various chromatin remodelers that slide, evict, and sometimes disassemble the nucleosomes. And these all come into play in creating an ordered array of nucleosomes that's optimal for transcription to occur properly. Okay, this represents some of what goes on when you're trying to get a gene transcribed. Okay, at the top we have the DNA sequence itself without the help of any proteins. And there is a preferred position depending on the DNA sequence to a certain extent, but this needs to be reorganized in order to get efficient transcription from this particular gene. So we're showing the binding of one general regulatory factor upstream of the first nucleosome, and then we're going to bring other protein complexes, SWI/SNF and the RISC complex, in play in order to move the nucleosomes around on the DNA template. Another set of remodelers includes the ISW2 and NO80 complexes, and these together collectively will create this phased regular array with the transcription start site correctly placed relative to the first +1 nucleosome. And these are necessary to, all these complexes are involved in generating this ordered array, which is optimal for transcription. So example of how we know this, like how can we know where the nucleosomes are? I'm going to give one example of a chromatin precipitation method, and that's going to tell us where the nucleosomes are in this particular, for these two particular genes. Okay, now this is one example of a more general type of approach that we're going to be talking more about later, more involving what proteins are bound where in the genome. So this doesn't have a protein antibody part to it, we're just looking at the DNA sequences in this case. But the principle is similar. We're going to use next generation sequencing, also called massive parallel sequencing, in order to relate the DNA sequence itself to the locations of the nucleosomes. Okay, the first approach is you're going to get chromatin, purify it, and importantly, digest it with an enzyme called micrococcal nuclease. Now, this is a general nuclease, and for naked DNA, it will just digest it all. But the DNA that's bound to the nucleosome proteins, physically bound to it, is much less sensitive to its activity than the linker DNA that's between nucleosomes. So we're going to use this trick to isolate the DNA that's actually physically associated with the nucleosomes and then characterize that. Because the DNA in the linker is sensitive, will be digested away, and what we'll get out of this is a core octamer with its bound DNA, and it's the DNA that we're going to want to analyze in this case. So here's a picture representing different forms of nucleosome arrays. So on the one on the left, we have phased regular array. and look at cell 1, cell 2, cell 3, cell 4, we can see that each nucleosome, starting with the +1, is located at the same sequence in each of those cells. So that's to say it's a phased regular array because there are regions of the DNA in all the cells which are not protected by a nucleosome, and regions of the DNA which are protected, and those are the same in each, thereby allowing us to identify them. By contrast, the other two, B and C or B, are different types of regular arrays that are not phased or irregular arrays. And you can see from that the nucleosomes are there, but they're located in different places relative to the DNA sequence. So they can be shifted over, and there can be regions with no nucleosomes at all. Now, the results of those particular arrangements predict different things about the DNA that we isolate. In all cases, we're going to cut the DNA, chew away the linker DNA, and then massively purify that, and then massively sequence everything we get. Now, in the case of the phased array, all the cells have the nucleosome located at the same place, so they're going to be DNA sequences which are not bound by nucleosomes in any of the cells, and those that are firmly in the middle of a nucleosome in the other cells. So that's in contrast with the other arrays where any particular sequence of DNA might be protected by a nucleosome, but it might not be. And so the result there is going to be a blur. Right, so we're going to isolate the DNA. All the pieces should be about the same size, 147 nucleotides. If the nucleosomes are precisely positioned, each piece of DNA will be associated with a nucleosome in a certain way. But if it's irregular, the pieces are going to be different according to the individual nucleosomes that were there. So this is where the sequencing comes in. We're going to get our nucleosomal DNA. We're going to sequence it massively, get millions and millions and millions of sequences, and then figure out where in the genome each one of those sequences came from. Because we can do that, we know what the genome sequence is. We just get our sequence, ask our computer to tell us where it came from, and then we're going to display that as a plot. So the millions of pieces are generated, sequence aligned with the genome, and this places each sequence according to where they came from. So this is represented as read counts across the gene, or genome as the case may be. All right, so if the nucleosomes are precisely positioned, we will see peaks that are centered on each nucleosome. If they are not precisely positioned, the reads will blur, and because all the DNA will be protected at least in some of the cells, if not others. Okay, so what that looks like here, in the case of the phased regular array, there are certain sequences which are not covered by nucleosomes, and those will be represented as troughs. And then we have peaks, where the peak represents the center of each of those phased nucleosomes. So the series of peaks tells us this is a phased array of nucleosomes, so we can see where the nucleosomes were relative to the DNA sequence. Okay, the figure on the right shows a case where there's no specific location for the nucleosomes, so there might be some preferences in some places. But the sequence you get doesn't really have peaks and troughs. It's kind of a blur over the whole region. So that's what you would see if you didn't have a regular array of phased nucleosomes. Okay, so for next time, we're going to save this, the large-scale three-dimensional structure of the chromosome and topologically associated domains. We're going to put that off next time.